Hey everybody, Mr. Murphy here. I'm going to be looking at Chapter 9, all about DNA. So the first part of Chapter 9 is about the actual structure of DNA, then its process of replication, and last 9.3 is about mutations. The question sort of to be thinking about throughout this chapter is what can we learn from ancient DNA? So how does our understanding of DNA today inform the past? So we've gone through this in class with the history of this process, but we'll go through kind of an abbreviated version of it. That in the early days of figuring this stuff out, scientists had established a few important things that were needed to be true based upon our observations about genetics and inheritance. And so DNA, we knew, had to be present in the nucleus and in chromosomes. We knew that chromosomes were somehow an element to this. And sorry, not that DNA had to be those things, but that whatever was responsible for our genetic inheritance of things had to have these things. It is a molecule that doubles in the cell cycle. It's twice as abundant in diploid cells, and it has the same pattern of transmission as the genetic information that is conveyed by it. So we first had identified this by Miescher. We talked about him in class. He isolated what he called nuclein, which would later be referred to as nucleic acids. So this was before we knew about double helixes and before we were confident that it was responsible for genetics. Here you can see some cells that have been stained, or the nucleus of cells, maybe. This picture is a little more compelling. You can see the actual chromosomes that have been stained. So there's different mechanisms by which this could be done historically, and there's a whole rich and very cool history to it. But an important outcome of experiments along these lines were that non-dividing cells have the same amount of nuclear DNA, and importantly, that after meiosis, gametes only have half as much. So that's pretty compelling data to demonstrate what we now know about how reproduction works and how each of the gametes has half a complete amount of DNA, and that's why the whole organism that results has one complete amount of DNA. So this fits sort of what we now take for granted. Here you can see just a little graph showing the vast majority of these cells having one amount of DNA. Some amount of these cells having a little bit more. Those are probably cells that were in the middle of mitosis. They've just passed S phase, so they had twice as much DNA in the cell. You can see the book for a little more info on those graphs and things. So we, in the history of figuring this stuff out, one of the things that was discovered is that bacteriophages, which are viruses that cause an infection in bacteria, they contain DNA and very little protein. And so when a virus infects a bacterium, it's injecting its DNA into that bacteria, which then is altering its genetics and sort of repurposing the protein synthesis and all of that. And so it provided evidence that DNA and not protein was the source of actual genetic material. And here you can see bacteriophages, which is so cool. I mean, it looks like this crazy little lunar lander on the surface of a cell. And you can see what happens is it injects its DNA. That DNA then begins to be transcribed and translated, and it builds new proteins that build new protein coats for new virus molecules until that cell literally just bursts with viruses. And that is the very scary but also kind of weirdly cool process by which viruses replicate using our cells. So transformation is a separate experiment which demonstrated that DNA from one strain of bacteria could actually be transmitted to another strain. And so this established that prokaryotic organisms rely upon other mechanisms to create some genetic diversity by literally just sending DNA out of themselves and into nearby neighboring bacteria. And so it creates this sort of hybrid or transgenic version of that organism, which is, again, kind of cool mechanism to increase diversity, especially considering that prokaryotic organisms don't sexually reproduce, and so they would otherwise be bound to sort of being exact clones of themselves. This gives them a mechanism to get a little bit of diversity, which helps with their continued survival. And so you can see the hypothesis set up here about DNA transforming eukaryotic cells. This will run through, in your textbook, it runs through the details of this, but essentially demonstrating how this could be achieved and how it was isolated and identified. It's a cool and interesting thing, but basically just demonstrating that yes, DNA can be changed. That the cell can respond in new ways if its DNA is altered. So there were a couple questions that still needed to be figured out. One was, how is DNA replicated between cell divisions? We know that it has to happen very quickly. We know it has to be very reliable. And the second is, how does it direct the synthesis of new proteins? And these are going to be sort of the overarching themes of the whole unit that we're doing here is first off, how does DNA do the things it does? And then what do we now do that we have this genetic information stored as DNA? And that's where the protein synthesis and all that good stuff comes in. So a whole bunch of different scientists involved here. Rosalind Franklin appropriately gets some credit in our textbook. She was one of the crystallographers who identified evidence that supported the helical nature of DNA. And here's a diagram demonstrating how the X-rays that were shot out, there's a lead screen, and so it's a very directed blast of X-ray that's allowed through and hits this DNA sample, and then that refracts onto a screen. And because when light passes through objects of different densities and different materials, it will bend its direction. It's like why rainbows happen as light passes through water droplets. So the pattern that very famously we see is notably the X-rays pass through the sample and then refracted in these different directions. And then based upon your knowledge of sort of what type of refraction would happen based upon the wavelength of the light source and the density of the material that it's passing through and all that kind of stuff, you kind of work backwards towards, okay, what does this mean we hit? And so, you or I probably look at this and we're like, that looks like a fun art project or something. I feel like my kids have a thing where you just like drop paint in and spin it and you get something that looks like this. But this of course was very meaningful data in terms of working backwards to figure out what does this tell us about what these molecules actually are made out of. And here's Rosalind Franklin. So additional other things we learn about is the structure of the different nucleic acids and the nucleotides that make those up, the different purines and pyrimidines, purines being A and G, adenine and guanine. And Chargaff was the one who identified that there's a pretty consistent amount between these, that whatever amount of adenine you have, you're going to have the same amount of thymine, and whatever amount of cytosine you have, you're going to have the same amount of guanine. So it's not necessarily all four are the same, but if you have whatever the amount is of A in a sample, you're going to have roughly the same amount of T. And so this became known as Chargaff's rule, and also supported then the idea that the double helix fits this fact here that we always have them paired up in a way. So it was another piece of evidence that would ultimately lead to Crick and Watson's double helix model, which demonstrated, again, using Franklin's X-ray data, demonstrated that DNA was in fact helical, that the two strands are antiparallel. And what that means is that they are oriented opposite to each other, and that's important when we think about then the enzymes and how they work upon them when we talk about replication. So here's that famous photo. They suggested that the nucleotide bases were on the inside and the sugar phosphate was on the outside. There were other individuals who had sort of hypothesized like a reverse version of that, but this was the one that ultimately stuck, and it's the one that we've since confirmed. Cool. So there's a few things just to note. It's double-stranded helix. It's right-handed. I'll come back to that in a second. And it's antiparallel. Already mentioned that. The other thing is that there are major and minor grooves. The major and minor grooves refer to like the spacing here, that because of the way in which it twists, it's hard to imagine this three-dimensional object, but because of the way it twists, you have one set of backbones where there's a smaller groove here, and then for the continued twist, you'd get another region that's a major groove that would be a little bit longer of a distance, and that's just because of how the bonding of these things is arranged, that as you twist it together you get this sort of pattern of major and minor grooves. The right-handedness is referring to the fact that if you were to take your right hand and sort of wrap it around this, so your fingers are wrapping upwards on this this continued spiral, and your thumb is pointing in the upward direction. If you were to try to do that with your left hand, you notice that your thumb is pointed in the opposite direction, then your fingers are twisting. And so it's not right hand, left hand, like meaningfully related to your hands. It's just referring to what direction does it spin. Because if you, I have my Connects model of this, and depending on if I twist it this way, I get one thing, and if I twist it in this direction, I get a different shape. Now, ultimately, it kind of looks all the same. This would be right-handed DNA, which all DNA on Earth is. But chemically, it is possible for the molecule to have instead been twisted in this opposite direction, which would be considered left-handed DNA, and that's just not what we see. And so there's a sort of evolutionary point there about that. And the reason why there'd be favored one versus the other is really just about the subsequent proteins that are evolved to work around and wrap around this thing. That being twisted in one direction versus the other just alters sort of how it can interact with other enzymes and things that are around. So it's an interesting point. It's not one that I'm going to dwell on or quiz you on or things. Okay. Same thing, the grooves is not a concept that we need to dive into. Just understand that if you are interested on a molecular biology level of some of the more details about this, that the chemical structure of DNA matters in just so many different ways in terms of how it interacts with the different components of itself, and then also all of the other things going on inside of the cell. So your book spends a little bit of time dwelling on that. Again, not the most pertinent to what we need to be concerned about. But here you can see sort of why the creation of these different grooves of the spacing, and why we get a different pattern to how this thing wraps up. All right. So DNA's four main functions. First off, storage of genetic information, and conveying that information between generations is very important. Also, for it to replicate during cell division, its structure is important to enabling it to replicate quickly and efficiently. It is susceptible to mutations, which might not seem like a positive, but that provides opportunities for genetic diversity. Too much would, of course, be problematic. Too little can also be problematic. And so the structure of DNA lends itself very well to providing the opportunity for genetic diversity through the process of mutations, while not overly exposing itself to harmful problems. And then the last one is the actual expression of that coded information. So this is where we then take that information and turn that into proteins. So that covers the broad overview of DNA. The next section will be all about replication, and let me know if you have any questions. Thanks.